Today my topic is lateral transport in plants. Lateral transport in plants. So, in lateral transport, so in there's it is learned that long distance transport of sugar like solutes takes place through sieve tubes. It is not relevant to think that always the translocation takes place only through sieve tubes. Sometimes the solutes also will move into lateral directions. For example, if radioactively labeled sugar is supplied to leaves towards one side in sunflower plants, the radioactive carbon containing sugars reach to seeds in the one radius of the flowers only by translocation in axial manner. But if one or two leaves, this is the leaf's internal structure, leaf, in one or two, in only one or two leaves, on the other side the removed with the radioactive carbon is supplied to seeds in all parts of the flower. From this, it is understood that by introducing sugars with radioactive carbon to leaves towards one side, depending on the conditions, they could be made to translocate to other sides of the stem also. So, this is the leaf internal structure. This is the in veins, small vein. So the in the cells, this one is the cell wall, this is the cell membrane. In this is the cytoplasm. So these are the mesophyll cells. This is the bundle sheaths. This is phloem, companion cells, parenchyma, sieve elements, companion cells. So, if the leaves are removed from the stem of the Y-shaped branch, the photosynthetically prepared sugars from the region with leaves will be translocated this partly to the sieve tubes in the regions of the branch without leaves in the lateral directions. The presence of plasmodesmata between the cells around sieve tubes and them, the solutes could be translocated laterally to the other parts in the continuous symplast system. A part of such laterally translocated solutes reach the bark. Sometimes a part of laterally translocated solutes also reach the xylem vessels and against may reach the sieve tubes. These factors affecting translocation: temperature, light, oxygen, moisture, stress, and metabolic controls. Temperature is translocated out of the leaf. It is sensitive to temperature. The out of the leaf is sensitive to temperature. Low temperature generally reduces translocation. High temperature increases the translocation process. Light intensity, which influences growth patterns of plants, affects the distribution of and translocation of metabolites. Low light intense, intensities with inhibit root and shoot growth, reduces translocation rates, particularly to the root system. The necessity of oxygen for solute translocation is particularly noticed at the time of vein loading. So, moisture, stress, translocation of solute, and plant growth are affected simultaneously by moisture stress. The assembly transport of leaf, it is especially sensitive to moisture stress. Transport movement within the vascular system is not much affected by moisture stress. Metabolic controls like other any physiological process translocation also required required metabolic energy requires metabolic energy. So respiration plays a major role in translocations, and all factors with influences respiration naturally affects translocation. So these are the affecting for translocation factors. First one is the temperature, light, oxygen, moisture stress, and metabolic controls. So temperature, it is translocation out of the leaf. It is the sensitive to temperature. Low temperature is generally reduces translocation. High temperature increases mm, increases the translocation. High temperature increases the translocation process. So light also, it is the influences growth patterns of the plants, affects the distribution of and translocation of metabolites. Low light intensities, which inhibit root and shoot growth, reduces translocation rates and particularly to the root system. Oxygen, the necessity of the oxygen for solute translocation is particularly noticed at the time of unloading. So moisture also affecting moisture stress. So the plant's growth are affected simultaneously by moisture stress. The assimilate transport out of the leaf is especially sensitive to moisture stress. So metabolic controls like physiological process translocation also requires metabolic energy. So some respiration plays major role in translocation. All factors which influences respiration naturally affects translocation. Source source sink relationship. Source sink relationships. So actively photosynthesizing leaves are considered to be the source, while all other organs utilizing carbohydrates, these are regarded to sink. The carbohydrate is transported from source to the nearest sink. Upper leaves on the plant, the export principally to the shoot apex, while lower leaves to the roots and middle leaves to the both. The translocation of sugars from source to sinks takes place through sieve tubes of the phloem. So, since phloem sieve connections are on both sides of the stem, the leaves on the one side may be more efficient at exporting carbohydrates or other assimilation to sinks on the same side. However, in many species there is extensive cross linking of phloem tubes, especially nodes at nodes, and this eliminates a one side transport of assimilates. During translocation from source of sinks, the sugars synthesized in the leaf cells move symplastically to the sieve tubes, and the sugar at the sink end is absorbed and either actively converted into cell constituents or to other carbohydrates, which have little effects on osmotic potential and hydrostatic pressure of the phloem. The loading of the sugar in the phloem at the source end and their removal or conversion at sink and establish on osmotic potential or hydrostatic pressure gradient. This and establishes on osmotic potential. So these osmotic potentials, for example, or hydrostatic pressure gradients. So this gradient causes the movement of water and sugars from the source to the sink. So the if the it is the increased photosynthetic activity in the source, cells may increase the rate of translocation to sink cells. However, sink cells must be able to utilize sugars efficiently so that the differences in the hydrostatic pressure is maintained. If they are unable to utilize the increased production, there would be steady buildup of sugars in the system, causing a feedback inhibition, resulting in reduces photosynthesis. The sink capacity will in fact determine the rate of photosynthesis in source cell. Assimilate uh, partitioning during vegetative phase. So leaves and other green tissues are the source of assimilates in plants. During vegetative growth, root stems or developing leaves or sinks, which may compete with each other for the assimilates. The proportion of the assimilate partition to these three organs can influences plant growth. and productivity. At this stage, the plants may prefer to invest more of assimilate to leaf area development so that it can capture more light. However, the leaves also require water, nutrients, so investment in root growth is also necessary. So sometimes some crop plants such a most cereals have essentially no stem growth during vegetative development and favor uh, partition to leaves and roots only. Thus, leaves acts as zinc only for limited period. They need energy and carbon skeletons for their growth and development until they produce enough assimilate to handle their requirements. In most cases, the leaves become almost self sufficient when 50% when 50% uh, when 50% their final area is achieved. Thereafter they become exporting and have as a source. So the fully developed leaf may export 6 to 60 to 80%. It is assimilate under good environmental conditions of photosynthesis. As the leaf gets older and begins to since it fails to support its own energy requirements, then it does not export or import assimilates. So the some minerals may be exported out of the leaf uh, before it is shed off. So during and assimilate partitioning during reproductive phase. During reproductive growth, most of the assimilate is translocated to flowers, fruits, and seeds. So, in the determinate spaces, the zinc becomes too strong that almost total assimilate is transported. Reproductive parts in these plants, leaf and stem growth cease- uh, ceases at flowering. However, in indeterminate spaces, the vegetative and reproductive growth may occur in simultaneously. Uh, in these spaces, if there is much vegetative growth, the reproductive yield may be reduced. In most annual seeds become a dominant sinks after their in initiations. So during seeds filling, the major part of the assimilates is used for increasing seeds weight. So source sink relationship determines the growth and yield of fruits and seeds to horticultural crop plants. The growth of the organs depends upon the mm, it depends upon the strength of the source and ability of the source organs leaf. Supply assimilation assimilates from the source and limitation on transport of assimilation either due to the long distance or high translocation resistance or competitions from other side sinks. So these are the assimilation mm, partitioning during reproductive phases, so source sink relationship, and affecting factors also affecting to translocation, and lateral transport in plants. So this is the lateral